because they, for the amazing work on residue analysis that they have been doing during the last few years. Julie from a bit longer, Emmanuelle from more recent, but these basically resulted in the publication of these two seminal papers that, sorry, I think I'm not exaggerating if I say that they revolutionized, in a sense, the world of prehistoric archaeology. So this paper published as a first author by Julie, first diarying in Green Sahara, Africa in the 5th millennium BC, which is now 10 years old, so 10 years anniversary for this paper. And then a much more recent paper published by Emmanuelle in 2020, accurate compound-specific C14 dating of archaeological pottery vessels. I think that these are two papers that can be also of inspiration for all of you. As you know, these seminars are organized in the framework of the teaching activities of the PASAP Med PhD course of the University of Bari, Aldo Moro, the Politecnico of Bari, and the Institute of Heritage Science of the National Research Council of Italy. And I mean, I think that is pretty something to say that these two amazing papers were the result of the PhD work that Julie and Emmanuelle carried out. So I think that for many of you, a lot of you are PhD students, students of graduate school, so it can be really of inspiration. So just I would like just to pass to the proper presentation now of our colleagues. So we will start with the first presentation of Julie Dunne. Julie is a biomolecular archaeologist, which is based in the Organic Geochemistry unit at the University of Bristol. She's an organic residue specialist, completing her PhD in 2014, based on the investigation of diet and subsistence practices of prehistoric groups in the Green Sahara of Holocene North Africa, using a combined archaeological, molecular, and isotopic approach. This work resulted in the publication in Nature, the one that I just showed to you, on the first direct chemical evidence for diarying in Green Sahara Africa during the 5th millennium BC. Following this, she worked on a 3-year Leveroom funded project called The People in the Green Sahara, exploring the economic, ecological, and demographic history of the enigmatic Green Sahara to address how Holocene climate change affected broad-scale population dynamics and the role of climate as a driver in subsistence change and cultural innovation. She has since worked on the NOC project, investigating diet and subsistence in West Africa, and as ongoing project in Central and East Africa. Julie also has a research interest in vessel use, combined ethnographic and residue analysis, and UK prehistory and the medieval period, and is currently working on pottery from post-medieval assemblages from Ireland. So thank you very much, Julie, for being here with us. And I give to you the floor. I share again the screen to present your PowerPoint.
Okay, thank you, Junio. Okay, so apologies for the technical hitch at the beginning there. But it's lovely to be here with you all in virtually. and I'm going to talk to you today about organic residues in archaeology. Julio, I can't see my presentation. I can just see a view of people. That's it. That's better. Yeah, I'm going to talk to you about organic residues in archaeology. What I'm going to do is give a run through of the technique. And then I'm going to present to you a couple of very different case studies to give you an idea of the potential for residue analysis on pottery and what sort of questions we can answer with it. So let me just move my own presentation on. Yeah. So what are organic residues? So there are a range of amorphous organic remains from a very diverse variety of natural sources associated with many different types of artifacts found at archaeological sites. So these can vary, for example, for soils and watercourses and middens. They could be objects such as mummies, whether they be animal or human, as you can see from this cat mummy on the screen. We can look at organic residues found in both skeletal remains and in bog bodies. There's a picture of a ship there that you can see. This is King Henry VIII's flagship. So King Henry VIII was an English king, probably the most well known as he was the one with 6 wives, 2 of which he beheaded. And his flagship was sunk just off the southern coast of England 500 years ago. And when it was raised 20 years ago, it was found to be covered in this very sticky, dark material, which organic residue analysis showed us was actually a pine pitch, which came from pine trees exported from Sweden, which told us effectively about long distance trading links in the medieval period. We can also look at art objects. So this picture you see in the middle of the screen there is a lovely little lion's head made of amber, which we identified from the organic residue analysis, which was excavated from a Bronze Age tomb in Katna in Syria. And we can also look at things like organic pigments, as I said, resins, tars, pitchins and bitumen, plant remains, but by far the most common artifact that we look at organic residues from is pottery. So next slide, please, Junio. And how we do this is we use what are called archaeological biomarkers. And these are ancient biomolecules that carry information concerning human activity in the past. And these molecules, you can see some of them along the side there. These can be linked to biological sources due to their characteristic structures. Now, we might look at certain classes of compounds preserved in organic residues, for example, DNA, protein. But what we mainly look at is lipids. And lipids are the fats, waxes and resins of the natural world. And what these chemical structures you see here give us are essentially a chemical fingerprint of the commodities involved. Next slide, please, Junior. So as I say, by far the most common residue we look at in our lab at Bristol is organic residues in pottery. And the reason for this is pottery is abundant, it's ubiquitous, found on, once it's invented, it's found on archaeological sites across the globe because it is very durable. On its own, pottery is diagnostic, so we can infer the age of pottery from stylistic traits. And Emmanuelle is going to be talking much more about what else we can do in terms of the dating of pottery. But most importantly for our purposes, it preserves organic residues. Next slide, please, Junia. So organic residues in pottery can come in 3 forms. Very rarely you might find actual contents, maybe solidified or liquid, as a container fill. This picture on the far right here is actually a tin, but this tin contained was found in Roman London, so it's around about 2,000 years old. And when it was opened, you can see the cream still in the pot. And I always think it's lovely that if you look in the lid, you can see where the Roman lady who last used this, what was found to be a cosmetic, a face cream, where you can see where she scooped some cream out to put on her face. And I like to think of her doing that. The second most common type of organic residue are carbonized or encrusted residues, which might adhere to either the interior or the exterior of a vessel. Not so common, of course, but they can be useful both for looking at the commodities that may have been processed in the pot, the last uses of the pot, and they can also be useful for radiocarbon dating, although we should remember, of course, that they could be prone to contamination. But by far the most common form of residues are those preserved within the vessel wall. And these are known as absorbed residues. So these may originate from a single use of a vessel. It might be an accumulation of individual use events in a vessel over its life history. And I would say the best way to sort of think of this happening is that if you thought of a basic terracotta pot and you put some meat in it and some water and boiled that up on a fire, what you'd see floating on the top of that is little globules of fat. And these fat globules are lipids. These are the chemical things that we're looking at, and these absorb into the porous framework of a ceramic over the lifetime use of a pot. Next slide, please, Julia. Yeah, can you go on 1, 2, 3, 4? Because there's pictures on there. That's it. Thanks. Okay, so how do we do this? So obviously I must emphasize that it is a destructive technique, but it is very minimally destructive. We really sample a piece only the size of about a euro coin, and we can return the remainder of pot sherds to you. And what we do first of all is clean the entire outside surface with a modeling drill, and that's to remove any contamination from either perhaps which occurred in the soil, or perhaps from handling during or excavation or fine processing. The small piece of pot is then ground to a powder, and it undergoes a solvent or an acid extraction to get out, as it were, the lipid biomarkers. Now, what we do, the first step is gas chromatography, and what that does is it literally tells us if there was anything processed within the pot, and it separates out any fatty acids or other chemical structures that we might expect to see, and gives us an idea. It quantifies the abundance of any chemical structures. Then our next step is to run that on what's called a gas chromatograph mass spectrometer, and what that does is it identifies the structures present in the vessel. Following that, we would run this to get the value of the delta 13C values of individual fatty acids, by far the most common compounds found in vessels. So next slide, please, Julia. So what archaeological questions can residue analysis answer? So there's a really broad range of questions that we can attempt to tackle. The most common is commodity use. So what types of foodstuffs might be processed in pots? So that can tell us information about diet. And I'm just going to talk in a minute about what sort of foodstuffs we can identify. Diet and subsistence, it might tell us something about technology and ritual use. We can also look at the relationships between form and function. So specific vessels might be used to process specific foods. A good example of that is the earthenware sieves that my colleague Melanie identified from 5th and 6th century Europe. And these sieves were found to contain dairy fats, suggesting that they were used as essentially cheese presses. We can also look at methods of cooking food, say, for example, boiling or roasting. We can look at the decoration or repair of vessels. We found British vessels to be repaired with a birch bark glue, and vessels may be decorated with similar kind of things like bitumen and so on and so on. We can sometimes look at post-firing treatments, and on a broader scale, we can look at animal management. So we might be able to identify, say, for example, pastoral management of animals, transhumance, and so on. And in the broad scale, we can use this to look at very broad patterns of diet and subsistence across temporal and spatial frameworks. For example, when does dairying begin in Holocene North Africa? As Julian mentioned, the Nature papers that was identified in Libya in the 5th millennium BC, and that does seem to be sort of the earliest that we've found so far. Dairying does happen in Sudan and so on. Roughly similar times, or perhaps even a little bit later, but we haven't seen any earlier evidence than that as of yet. We can also use, a broader question is, could we identify changing patterns of food consumption by looking at different vessel types across long durée timescales? Next question, please. Next slide, please, Julia. And so the sorts of commodities that organic residue analysis can identify, well, by far the most common commodity that we do see in pots are terrestrial animal fats. So we can differentiate between ruminant versus non-ruminant animals. So if we were looking at European prehistory, for example, we would that would either comprise ruminants, cattle, sheep, or goat, versus pigs. But that becomes much more complex in a context, for example, such as Africa, where there are very, very many diverse types of wild ruminants found, say, for example, in Holocene North Africa. So it's a much more complex question there. So we can differentiate these carcass, the animal fats that people might be eating, from dairy fats, and that's obviously vastly important in terms of identifying the origins of dairying. We can also use specific biomarkers to identify the processing of fish, including shellfish and marine mammals. So, for example, seal processing was identified at Kastilberg in South Africa, and this. It has to be emphasised that this is always carried out, as part of multidisciplinary studies. So we would want to investigate what animal bones were found at the site, and that will give us a much better idea of, we can tie the lipid results together with the animal bones. So we knew at Kastilberg that it was seal because the faunal assemblage was overwhelmingly seal. But where lipid analysis has a massive advantage is if there are no animal bones in the soil, such as Takakoi in Libya, where there are very, very few animal bones, then the lipids are acting as a proxy for animal exploitation. So we can also identify plant oils and waxes, and I'm going to talk a bit more about that in a minute. Beeswax. So that beeswax is a proxy for honey, the exploitation of honey by ancient peoples, and it would be a very rare source of sweetener for them. And then finally, we can look at processing of resins, tars, and bitumen, often for technological purposes. So next slide, please, Julia. So I won't go too much into this, but just to give you a little overview on distinguishing animal fats. So this plot here, as you'll see, there are ellipses for ruminant dairy fat, ruminant adipose fat, and a porcine adipose fat here. And we know from the analysis of large numbers of modern animals, which obviously act as a proxy for the animals exploited in the past. So we know from analysis of modern animal fats that fats from those particular animals plot separately, and this is based on the isotopic difference between the C18 in ruminant carcass and milk fats, for example, in the case of differentiating carcass and milk. And that's because there's a more negative delta 13Z value for milk fat C18 fatty acid than there is for body fat. This picture, which I won't comment on much, but does become more complicated in when we get into what sort of forages the animals might have existed on. For example, we know that in the UK and Europe in prehistory, animals would only have been eating C3 plants, but in Africa, for example, they might also be eating C4 plants, and this is where lipid analysis can also be very helpful in that it's identifying the types of plants that animals are eating, and thus providing an environmental signal for us. Next slide, please, Junio. So, and yeah, you can go on to the next one. So, as I said, I'm going to do two case studies. So this is something I've just been working on recently, and this is the identification of plant lipid biomarkers in archaeological pottery. So next slide, please. So the detection of lipid biomarkers denoting plant processing remains quite rare in comparison to the identification of animal fats. And that's despite the fact that the invention of, for the first time, thermally resistant cooking vessels, so ceramic pots, this was a crucial step in human technological progress, and this fully unlocked the potential of plants as foodstuffs. So it allows an increased dietary diversity, a greater variety of nutrients, and more stable and palatable foods. So, for example, there are many foods that need to undergo prolonged boiling to detoxify them, and that's something that only became possible once pottery was invented. Now, the reason that the identification of plant processing is quite rare is because that if meat and plants are processed in the same vessel, which is sort of a logical enough thing, people would very often probably eat mixtures of meat and plants, maybe in the form of stews and so on, then meats which have a much higher lipid content will swamp the plant signal and make it very difficult to identify. So we have, however, found plant lipid distributions. Essentially, the oldest known so far were found in at the site of Takakoi in Libya, and there was evidence for processing of leafy plants and also aquatic plants and plant oils as well. And that bore out the remarkable exploitation of plants in that area, where we saw identification of amazing basketry and so on made from plant material. We also have seen some identification of plants in the Moroccan Rift region and East Africa. So there's a lot of potential for identifying plant processing in African pottery. So next slide, please, Junio. So we're going to look at the knot culture in West Africa, investigated from, over 15 years now by the Knot Research Project at Goethe University in Frankfurt and the National Commission for Museums and Monuments of Nigeria. So the knot culture, which is known for its remarkable terracotta figurines, and some of these are not easy to see from this, but some of these are you know, massive. They're really several feet tall and amazing sculptures. I think the oldest known terracotta figures possibly in Africa. And the culture dated between 1500 BC and the last century BC. Also, it's also known for some of the earliest evidence for iron production in West Africa. And the key study area of the project lies in the south of the Kaduna Plains, covering an area of around about 300 kilometres squared. Next slide, please, Julia. So the reason I really got involved in the project was that reconstruction of the diet of knot people was very, very difficult, because the acidic soils meant there was a total lack of animal bone and very poor preservation of plant material. What we really found were carbonized remains in the knot plant record, and we can surmise that that probably gives an incomplete picture of food acquisition and preparation. So there's a distribution map at the top there, showing the I think it was 10 sites that we sampled pottery from for this study. So lipid residue analysis of knot ceramics provided the opportunity to determine what animals and plants and knot people may have been exploiting. Really, the only avenue left to us, bearing in mind the scarcity of faunal and archaeobotanical remains. So next slide, please, Junio. So we sampled and analysed 458 potsherds from the 10 sites I mentioned. There's a picture of Gabby. I'm sure some of you know Gabby. You know, documenting the vessels excavated a couple of years ago. This was the lipid recovery rate was pretty low at 15%. And that's a complete contrast to pottery from, say, Takakori, where recovery was virtually 100%. The preservation was so good at that site. And that does highlight really the differences that you can see in pottery assemblages. That is really the kind of bookends, the low and the high. If recovery rate is low, though, the solution to that, it is a numbers game, and you know, you just have to analyze more pottery essentially to get a meaningful statistical result. So I was quite surprised once we'd done this analysis that there was 3 very distinct lipid categories, lipid profiles that arose from this work. So 1/3 of them, just under 1/3, comprised evidence for degraded animal fat processing. That's not published yet, but I will say there was no evidence for the exploitation of dairy products there. That was all carcass fat processing. Most amazingly, we found evidence for beeswax, and thus we surmise honey processing in 1/3 of the vessels. And this is interesting because even today there's a vast amount of gathering of wild honey. It's not a managed process, but people exploit wild honey from the rainforest, as they have done probably for hundreds, if not thousands of years. And then finally, the last set of profiles was a very vast and diverse range of plant lipid profiles, the broadest range I've yet seen anywhere in plant in vessels used to process plants. So next slide, please, Julia. Next slide, I see. As I say, very, very vast and diverse range of plant lipid profiles, comprising particular combinations or series of long chain lipids, so including even numbered alkanoic acids, so fatty acids, C20 to 32, odd numbered alkanoic acids, in high abundances, which is very unusual. Even numbered ones are indicative of plant vegetable epicuticular waxes. But it's very unusual, if not unheard of, to see high numbers of odd-numbered fatty acids. Sequences of alkanes, again indicative of epicuticular waxes, and then a series of diacids of high carbon chain length. So, as I say, some of these were not previously seen in archaeological vessels. They look quite complicated, and I won't spend a lot of time sort of talking about them, but if you see from the top left-hand one, maybe right-hand one your side, so NOC 090 is a profile which just comprises, the IS means internal standard, so that's how we quantify, work out the abundance of what's there. And this just comprises, for example, 3 long chain N-alkanes, the C29, the C31 and C33. And this is the first time we've literally seen an archaeological vessel with a signal like this. And N-alkanes are indicative of epicuticular wax processing. So that's the shiny surface of leaf waxes that you see when you look at a plant. So clearly that tells us that in that pot, a specific plant probably were being processed essentially, and they are, and it indicates the processing of leafy plants. Various other combinations here. So if you look at the ones with the blue squares alongside the those with the red circles, so the red circles are long chain fatty acids, and the blue squares are the diacids. Now we've seen the processing of diacids in pottery at Takakori, but in Libya, but in the short chain, and these are indicative of triangles. Here, these are indicative of processing of diacids. And these distributions suggest a possible processing of various plant types in vessels singly or in combination, with low lipid concentrations typical for plant processing. And I've just seen the time, so I've got to speed up a little bit. So where do these profiles come from? Well, we know from the archaeobotanical remains that pearl millet and cowpea was cultivated at NOC settlements, and pearl millet's the only C4 crop documented in the region at the time, and the enriched delta 13C values from from some of the pots suggest a C4 origin. So we did purchase some modern day millet and cook up in terracotta pots in the region, and we found that the delta 13C values from these pots was in very good agreement with some of the values from the NOC pots, suggesting that some of the C4 lipid signal in the pots derives from the cooking of pearl millets, which is in very good agreement with the archaeobotanical remains. Next slide, please, Julia. So, as I mentioned,
Julie, take easily your time, no rush. And can you please all switch off your mic, please? Thank you.
Okay, if you're sure, I was speeding up a bit there. Okay. Yes, okay. Still next slide anyway. So another type of plant, next slide, Julia. Another type of plant that we find evidence for processing, or types of plants, are what are called traditional leafy vegetables or leafy greens. And these are indicated by the presence of the N-alkanes that I mentioned and the long chain fatty acids. So there's a vast range of these available for consumption in sub-Saharan Africa today, with over 1,000 species identified. And this includes indigenous African plants such as jute mallow, or bush okra, African eggplant, and bitterleaf, together with introduced vegetables such as amaranth, which are very often consumed in sauces. So fresh leaves might be used as what they call pot herbs, or vegetables in soups and sauces, while dried leaves are often widely used as condiments and flavourings for sauces and cooked cereals such as pearl millet or sorghum. So next slide, please, Julia. And what we found was really interesting was that leaves can be used in various ways. So they could be either boiled fresh, or quite often they're pounded and dried and then kept until the dry season when fresh leaves are scarce. Sometimes they're ground into a paste and added to sauces to thicken and give particular tastes and aroma to dishes. And one of the most important leaf providing species is the baobab, which together with other things such as okra, jute mallow and so on, they're cooked in soups with a little potash, and this gives a high mucilage content, or what's known as a slimy consistency. And this source is really common in high yam producing areas, because pounded yam is really known to be, which is possibly quite dry and hard to get down, as it were, but it's complemented by sources of slimy consistency, and used also with other cereal and tuber dishes. And this obviously, these leaves are it's a source of very good nutrition, essentially. So next slide, please, Julia. So other plant parts may have been processed in the pots. So for example, the locust beet tree, Parkia biglobosa, the leaves are boiled, mixed with cereal flour and eaten as a vegetable. The seeds, pods and fruit pulp are used in cooking and making drinks, and the seeds are also used as a spice, and that's very commonly used across West Africa, up to about 80% of all meals. And it is remarkable. It's a tree that virtually every part of it can be and is being eaten in some form, either as in a meal or in a drink. And this food stuff called sumbala, which you can see a picture of here, the seeds are boiled and then washed, boiled again, and left to ferment for 72 hours and then dried. So these are a very valuable staple. They would preserve for quite a good period of time because they've been fermented and then dried. And they have a very high fat content and astonishingly a protein content higher than many types of meat. It's a very good food source. So and we know that Parkia is found in knot period vegetation in the wood charcoal assemblages. So we might surmise that possibly was exploited by knot people. Next slide, please. And also present, we think, in these pots were underground storage organs. So 4 of the vessels, as you can see from the plot here with the green lines on the foreshortened chromatogram, we're able to identify these diacids and isotopically measure them, which is to my knowledge the first time that's ever been done, and they do suggest a very C3 origin, which yams generally are. So this strongly suggests the processing of plant organs, underground storage organs, which contain suberin as a major component. And the identification of long chain diacids is very unusual, and those with long chain lengths are considered diagnostic for the presence of suberin, which is found in abundance in the outer parts of underground plants. So yam was domesticated around about 2500 BC in this particular area, which does suggest a sort of a good agreement really with the processing of them in the pots. And yams are a great staple in West Africa. They're used to make something called fufu, which is a very thick, starchy paste, commonly served with meat or vegetable stew. And that can involve the peeling and subsequent boiling and pounding of the yams, although the yams are sometimes processed without being peeled, which is how we might be identifying the suberin from the outer surface. So in conclusion, next slide please, Julio. Yeah. So the plant lipid profiles fall into at least 7 and likely more distinct categories. And the presence of the diacids, alkanes, and sequences of long-chain fatty acids suggests that underground storage organs, possibly yams, together with leafy plants and pulses, were exploited from at least mid-2nd millennium BC, providing confirmation of the early origins of West African plant cuisine. And that confirms for the first time the importance of such plants in the knot diet. These might have been routinely prepared and mixed, probably different plants mixed in different vessels, and that's probably why we see so many different lipid profiles. And so this has allowed us to go beyond the identification of meals thought to consist mainly of meat and starchy plants, but we can now confirm, based on the highly diverse range of lipid profiles that we see, the preparation of ancient meals combining vegetables, pulses, underground storage organs, and possibly herbs and spices. Next slide, please, Julia. So this paper is available. If anyone wants a copy, do let me know and I'll send it on to you. Okay, so Julia, I did start a bit late, but I have got a second case study. Am I okay to carry on?
Yeah, please do it, Julie. Please do it.
Is that okay? Worry. Okay.
Yeah, absolutely.
Okay, so I thought we'd go somewhere totally different for the second case study, and this is something really just finished working on, organic residue analysis of funerary vessels, and I thought you would be interested in this because it's from 2 Romano British cemeteries. So next slide, please, Julia. And it does give us a completely different picture of the use of vessels. So in England.
Scusate, sorry, can you please switch off your mic, please?
In England, in Britain, common
se ci hanno messo cose che non possono entrare nei porti, come possono? Via terra dall'Ucraina portano via terra.
Scusa un attimo, sorry a moment, Julie.
Yeah.
Okay, it seems that they should all be. Sorry that I cannot deactivate myself because I'm dealing with your presentation.
Yeah. Okay. So, a common feature of burials from the late Iron Age to the late Roman period are sets of grave furnishings. And these are found, as you can see from this picture here, at Epinoy in France, not just in Britain, but across the empire. You find these suites of ceramic vessels, which we associate with food and drink consumption or storage, and they might include a range of dishes, platters, dishes, beacons, flagons, jars, and amphora, although they vary considerably in the numbers and types of vessels across burial to burial. And sometimes we can see single vessels or up to collections of 50 vessels in high status dining sets, as this image shown at Alton in Hampshire. Next slide, please, Julio. So the questions we might ask of these vessels is, do they represent a table setting where a meal is set out for the deceased in connection with the Roman institution of the funerary feast, the Silicernium, eaten at the grave? Excuse my pronunciation if that was bad. So classical sources discuss the importance of food and drink at several stages in Roman funerary rituals, although that documentary evidence is not directly applicable to the Marno-British context. But there's lots of evidence for the consumption or deposition of food and drink as part of funeral processes and rituals in Roman Britain, including animal sacrifices as parricide rituals, placement of food offerings during burial, and the consumption of food during commemorative visits to the deceased. So as I say, we also see these across the empire. At Southwark in London, a female cremation burial contained a very impressive array of plant remains, including typical Mediterranean species such as date, almond, fig, and pine nut. And that certainly tells us that there must be a movement of these commodities across the empire. Also common in burials are partial or complete animal skeletons. So we find, for example, the Eastern Cemetery in Roman London, a pot containing a chicken. We also see pig and goose. But what we must remember is that some products will not necessarily be visible archaeologically. So, for example, meat served off the bone, bread, cakes, fruit and vegetables, these would all degrade over time and not be present, including liquids such as wine and oil. So the question of these, the presence of these accessory vessels raises a question as how pottery was acquired for funerary use and why specific vessels were chosen. Were they made especially for funerary purposes, or were they recycled from the domestic sphere? May they have belonged to that particular individual, for example? And did they contain food for the deceased, or possibly to make their final journey? Or did they simply represent a symbolic meal? So we carried out organic residue analysis of two sites, one in Canterbury and one in a place called a fort called Baggington in Coventry. So next slide, please, Junior. Okay, I already talked through that one. So next one, please. Yeah, so next one, please. I obviously didn't say next ones. Yeah, oh, so sorry about that. So the recent excavations in Canterbury identified a small inhumation cemetery, which you can see here on this image, containing about 20 individuals. It's since been found to be a part, although a separate part of a much larger set of burials. But the cemetery was contemporary with an extramural shrine or temple, which was situated very close by to this small cemetery. And its dating of the vessel suggests it was in use from the late 3rd to late 4th or early 5th century AD, so quite late. Next slide, please, Junior. So a number of features, including, as you can see, the unusual depth of some of the graves, suggests that it might have been for individuals of high status. So priests in charge and their families, and actually I can confirm that aDNA analysis was done on the 20 individuals, and there are familial relationships between some of them. A large quantity and range of grave goods was retrieved from the burials. So examples included grave 6 of a female, which contained a glass vessel, a wooden basket, a brooch, a naturally rounded pebble, and a boar's tusk held within a cloth bag. Next slide, please, Junior. So this is a picture of the vessels excavated at Rodistown. So 11 of the graves contained between 1 and 3 very well preserved ceramic vessels. So there were 20 complete vessels. You can see them here. So I had slight kittens when these first arrived in the office, because the question was, can you examine them all? And I was like, eek, I can certainly sample some of these, but I was terrified at the thought of these lovely little funnel neck beakers, if you can see them along the bottom here, because we can, carry out sort of specialist sampling of complete vessels, but we wouldn't be able to get inside the funnel neck beakers. So those we didn't do, but the rest could be analysed. Present at the back is a really unusual face beaker, which has got a face mask, as you can, I think, hopefully just see there. And it's unique. It's the only vessel recorded from a grave anywhere within the Roman Empire. And as I say, present were 6 finely made funnel neck beakers. And what was really unusual about this is, as you can see, is some of these were quite small. 11 had rim diameters of up to 100 mil. So you can see some of these, they look almost like sort of miniature versions of bigger cooking pots. So next slide, please, Junia. So of the 20 vessels, as I say, I didn't sample the funnel neck beakers, but the rest were sampled, and we have a modified sampling procedure to minimise destructive sampling. So we sample inside the vessel so that you can't see that on the outside. So the lipid recovery rate for these vessels was excellent, 87%. So 13 of the 15 sampled yielded lipid profiles, suggesting they were used. So that's our kind of first interesting piece of evidence. So next slide, please, Julia. So these are 6 of the typical chromatograms from the Rodistown pots. Now, so you'll see they look vastly different from the plant lipid profiles, which I showed you from the knot culture. Much more simple, much less complex. They're all dominated by what you see here as the C16 and C18 fatty acids. And these are absolutely typical of degraded animal fats. So 5 of these vessels contained high concentrations of lipids, and that suggests that they underwent sustained use in processing of high lipid yielding commodities. And the remaining 9 had low concentrations of lipids. So these 5 were very likely used prior to deposition in burials, but it's quite possible that the remaining 9 weren't. So what you also might see here from looking at the profile of, so CRV07 there and CRV12 down the bottom there, 4 contained a series of long chain fatty acids and N-alkanes. I mentioned those earlier, and these are indicative and origin of leaf or stem epicuticular waxes. So that's consistent with the processing of plant material, likely leafy plants or possibly plant oils, together with animal products within these vessels. Next slide, please, Julia. So here we've measured, carried out GC IRMS isotope ratio mass spectrometry on these 14 samples. And as you'll see from the bottom box, as it were, 10 of the vessels plot in the ruminant dairy region. So 10 of these pots were used to process dairy, milk or dairy products. Although 3 of them plot at the range between ruminant dairy and ruminant adipose, suggesting some mixing with ruminant carcass products, and that's quite common. 3 vessels were used to process ruminants, so cattle, sheep or goat carcass fats, and 1 pig fats, as you see up there. Next slide, please, Junia. So let's concentrate on the meat first. So the 3 vessels which contained meat products, cattle, sheep or goat from ruminants, included the unique face beaker. And the lipid concentration from the face beaker was very low, suggesting it hadn't been used for cooking or processing food products previously. Now, suggesting, so it may have been used for decorative purposes before, or maybe it was made especially for funerary purposes, we don't know. But meat was presumably then left in the vessel as an offering, but we'd have to think that as the opening is quite limited to that vessel, it couldn't contain large chunks of meat or anything, they wouldn't fit in it. So we could surmise that this possibly was in the form of some sort of prepared meat stew or gruel or soup. The other two vessels containing meats had high concentrations of lipids, so they were probably be used before, presumably as everyday cooking pots. And similarly with the vessel used to process pig products. Next slide, please, Junio. But it's the dairy results which dominate. 5 of these had very low lipid concentrations. So again, these may have been made especially for funerary purposes. 4 of them were small or miniature, suggesting that these 4 of these small or miniature pots may have been made specifically for burial purposes. But another of the remaining 4 displayed very high lipid concentrations, so probably came from the domestic sphere. And although these were not clearly not made for funerary purposes and were used on a regular basis, again, their small size suggests it's unlikely that they were used for cooking. I think this is still a question, you know, a really interesting question that I'd like to sort of understand better what's going on with these small or miniature vessels, which are found in quite a lot of burial assemblages in Britain. I'm not entirely sure about, but across the rest of the empire. So what might be being held as offerings or processed in these pots? Next slide, please, Junio. So the dairy signal could come from cheese, and the Romans either made a soft curd or green or cottage cheese, which was eaten fresh, or it could be a hard, longer-lasting cheese. We know that many Roman writers, Pliny the Elder, Columella, wrote on cheese manufacture and the culinary uses of cheese. Milk from cows, sheep, goat, horses and asses was used to make cheese. And it was very popular Roman foodstuff used for the army. And in the Emperor Diocletian had to actually fix a maximum price for cheese. It was so popular. Palladius actually wrote a treatise on Romano-British farming in the 4th century AD, including a description of and advice on cheese making. So that tells us how important it clearly was in Britain anyway. Next slide, please, Junio. So 4 of the vessels which contain dairy products also contain this evidence for plant lipids. So this could originate from cheeses flavoured with herbs and other plants. So Columella discusses the addition of fig sap and pine nuts, both to coagulate the curd when you're making cheese, but also to give a pleasant flavour. And we also know from other writings that thyme and various other herbs were crushed and sieved into milk used to make cheese. Other sources of the dairy product signal could be butter, sour or sour milk products called oxygala, literally sour milk. This is sort of kind of like a yogurt. And something called malka and schistin, which we understand to be used as a medicament. Next slide, please, Junio. So then come on to the next site, which is this is a reconstruction of a fort at Baggington, and this is a very early cremation cemetery situated next to this fort here. It's one of the most significant Roman sites in the region, and constructed in around about AD 60 to 64, possibly as a response to the Boudiccan revolt. Graves contained 1 or 2 complete vessels, and so that we sort of investigated those found across the cemetery. Next slide, please, Julio. There's one particularly important one, which comprised a male 46 or older, buried accompanied by 23 vessels, which you see here. And these accompanying vessels were carbonated cups, a glazeware bowl, possibly central Gaulish, miniature flagons, an oil lamp, and a collection of Samian vessels, which were of Aretine origin. And these are very unusual in England at this time. and combined with the Gaulish and Spanish finewares in the grave suggests the individual was probably not just of high status, but also an immigrant from Gaul or the Mediterranean. Next slide, please, Julia. There was, oddly, but usefully, I'm going to tell you why in a minute, a few Anglo-Saxon features on this site, sunken featured buildings and graves. And one very small Anglo-Saxon vessel was found. So we analysed that just to sort of see, you know, what it looked like. And it actually contained a very high concentration of lipids, which confirms that the preservation conditions at the site are conducive to the survival of lipids. And this vessel was actually used, as you can see from the plots here, it was specialised, used in processing or containing ruminant dairy fats. I still think it's intriguing that, so this is a miniature vessel. Why are people making and using such small vessels to process foods, make dairy products? I don't know the answer to that. Next slide, please, Julia. So we analysed 17 vessels from Baggington, and all of them, except for the Anglo-Saxon vessel I've just mentioned, did not contain any lipids, so either just trace or very low concentrations in lipids. There's a complete lack of evidence for either storage or processing of foodstuffs in any of the vessels there, which suggests possibly that funerary ritual at the Lunte did not include, in contrast to Canterbury, the leaving of food offerings or meals for the dead in vessels. Although, interestingly, pig and possible chicken bones were found in grave 11, suggesting at least some feasting at the pyre. Next slide, please, Julia. So the results from Baggington suggest from the quality of manufacture, the pottery specialist thought that some of them may have been seconds, perhaps produced by the Lunte potter. And so bearing in mind that they were imperfect, it meant their status as damaged goods meant that it was expedient to use them in burials rather than, if they felt they couldn't sell them because they weren't perfect, but it would be okay to use them in burials. And this does suggest correlate well with arguments that, that pottery might have been specifically purchased at the time of the funeral, perhaps at markets or stalls set up near cemeteries, or even from burial societies, which may have kept a supply of potter's seconds or rejects, or even that they may have been disinterred from earlier funerals and reused. Next slide, please, Julia. There are certainly many examples of the use of old or repaired, imperfect or damaged vessels in burials at many sites in Britain, Kent, Ospinge in Kent, Silchester, Welling, Skeleton Green, Westminster Street in London, and St Pancras, Chichester. For example, of the 200 mostly complete vessels recovered at the large East London Roman cemetery, 23% were damaged in some way. So while this doesn't necessarily mean that vessels were not previously used for storage or cooking or other functions, it does suggest that when choosing vessels for burial, sometimes at least damaged pieces were frequently selected. Next slide, please, Julia. So in conclusion, some domestic pottery was clearly moved to the funerary realm. Whether those vessels belong to the individual in question, obviously we don't know that, but it's possible. Other vessels seem to have been purchased for use as funerary vessels, and seconds or wasters were used in some instances. There's certainly a variability in burial practices between the two sites, the chronological spread, but we would really need to do analysis of of many more sites. It would be interesting to look at more sites across Britain and of course across the empire, to see if we can get some broader understanding of burial practices. This is the first identification of dairy products. There was food offerings. They couldn't have been identified previously because they wouldn't survive archaeologically. Whether they are cheese or some sort of sour milk product, we don't know. But in some cases, the dairy products were mixed with leafy plants, presumably to enhance the taste of the foodstuffs. Meat was also deposited in vessels, possibly as prepared meals. And as I said, there is still some interesting questions, I think, about the small or miniature nature of some of the vessels. So I think that's the end of, I know I've gone over a bit, sorry, Emmanuelle. But I hope nobody minds. It was kind of nice to present the 2 case studies to you. There's a couple of papers there, and also the Historic England guidance to good practice in organic residue analysis, which I can send anyone if they're interested and want more information on residue analysis. Okay, thank you. I'm ready to stop talking.
Thank you so much, Julie, for this enlightening presentation, really so rich. I'm sure that there will be a lot of questions, but I think we can postpone them at the end of the presentation of Emmanuelle. So I'm going to present her. Emmanuelle, if you want, you can start sharing your screen meanwhile. So Emmanuelle Casanova did her undergraduate studies in chemistry in France, University of Lille. Then she moved to Bristol in the same group of Julie, who is with Richard Evershed, to perform her PhD with Richard, who was her supervisor. During her PhD work, she used the lipid residue analysis to reconstruct ancient diets during the Neolithic period, and she also worked on the development of a new method to radiocarbon date archaeological ceramics using their lipids residues. She graduated in 2019, and then she continued the research again in Bristol in the framework of a couple of 2 postdoctoral positions in order to apply and expand the new method she developed on other materials and archaeological questions. She then obtained last year a Mary Squoska Curie action, an individual fellowship to work at the National Museum of Natural History of Paris, to work on the paleodiet and chronology of Southwest Asia Neolithic. So thank you so much, Emmanuelle, to be with us, and I give you the floor.
Okay, so you can see my presentation and hear me well?
Yeah, perfectly.
Yeah, okay, so we're gonna start. So I'm gonna talk about more about the chronology we can obtain thanks to the ceramic vessel, but particularly from the lipid residues we find in the vessel, like Julie presented before. So the question we can first ask is, why do we need to date archaeological pottery vessels by the radiocarbon method? So first of all, the potteries are the artifacts we recover the most from the Neolithic period. They are really abundant at many archaeological sites. And the relative dating of pottery is one of the first steps to identify a cultural group. And the archaeologists use the typology and seriation study to really relatively date this ceramics. So I have put here some pictures of different ceramic assemblage for different periods. We have, for instance, here pottery from Alsace in France in the Neolithic. We have also in the middle pottery from the Neolithic period in Iran, and here some Roman pottery in the Rhone in France. So we can see, just looking at them, they are really different, and really just by the shape and decoration we can know where they're from and relatively how old they are. But in order to have absolute calendar age on this ceramic, it's a bit more tricky. And we can, if we can actually date the ceramic, we can go to have absolute dates on sites where there's no other archaeological artifacts. There's a lot of sites where the organic matter preservation is really poor, and there's no bones or charcoals or seeds that can be radiocarbon dated. So for this site, we need some alternative method to obtain absolute dates. So dating ceramics will be one way to obtain dates for such sites. So first we can wonder which carbon fraction we can date for pottery vessels, where the carbon is from, and if it's a reliable source of carbon for dating. So first we have carbon that can come from the temper of the ceramics. So that would be used during the manufacture, such as plants, for instance, or sand. And the event dated there would be the manufacture of the pot. But we need the temper to be organic. So usually if you have plant temper, this can be dated, but you also need to isolate it well from the clay matrix to obtain reliable dates for radiocarbon. The problems we can have with such tempers is when the pottery are fired, this temper will not be preserved, and then it won't be possible to use such as a carbon source. So I've put here picture of a ceramic where you can see some lines which are actually old from plants, but they did not really survive the firing, but we can see there was an organic temper in such shirt. So if the organic temper survived, this would be dateable, but it's really limited cases for this source of carbon. Then we can have the carbon coming from the fuel. So when the pots are put on an open fire, we can have carbon from the wood that can deposit on the surface of the vessel. So we would be actually dating the period of use of the vessel. And again, we need these residues to survive, and they're usually contaminated when the ceramics are curated, when there's like glue used or varnish, so conservation treatments in general. And actually, we won't be dating anything related to the vessel, but the wood that has been burned, and we can have issue with what we call old wood effect, so dating something that is actually older than what we want to date and is subjected to contamination because it's on the surface of the ceramic and is open to in the burial environment. So this is something we need to take care to be careful with. And there's also the visible organic residues that are from the cooking itself, like food crust. So we would be dating the use of the vessel. But again, this kind of residues need to survive and no conservation treatment applied to the ceramic. And again, it's subjected to the contamination in the burial. So these surface residues are used usually to radiocarbon date pottery vessels, but they are not necessarily really good for dating, especially if these residues have not been characterized before dating. And so we don't necessarily know what we actually date with those residues. So it's a bit tricky to work with that. Usually people try to avoid working with charred residues if they have bones or charcoal, something else to date. But if this is the only thing surviving, this is what is going to be dated. But yeah, I want to give a warning and be careful with such dating. And there's another source of carbon, which are all the lipid residue preserved within the vessel walls that Julie told you about. So we can find a wide range of these residues that are mainly animal products or botanical products in different forms. And the ones that we find more often in the pottery vessels with the highest abundance are the animal products, either terrestrial or aquatic resources, which are the target of my analysis. I mainly work with animal products for radiocarbon dating. And what I'm more interested in are the animal fats from terrestrial animals. So when we find them in pottery vessels, we will mainly have the C16 and C18 fatty acids that come from the degradation of the triacylglycerol in the archaeological context, and with a stable carbon isotope analysis, we can characterize them well and find out if they were porcine, ruminant adipose fat, ruminant dairy, and we can even distinguish aquatic resources with this information. So we have really good information, good characterization of those residues, and we know if they are reliable for dating, which is a really nice source of information we have here. So, like I said, I'm focusing on the dating of animal fats, but to be sure we won't have any carbon that is from contamination. I actually want to isolate these two fatty acids that are characteristic of animal fats, because these two compounds, I'm sure they are archaeologicals, but all the other compounds that can be extracted with them in the background, they can actually be not archaeological, and contamination from, for instance, plastic from the storage of pottery in plastic bags or handling post-excavation. So we really want to go for single compound radiocarbon dating rather than bulk organic residue dating, because a lot of study, well, quite a few studies have tried to date the bulk residues that we extract from the ceramics, but it never worked well. So this is why we need to go for more precise dating with single compounds. And the nice thing with working with these two compounds is we can have actually two radiocarbon dates for a single pottery vessel. And because they are contemporaneous, we should obtain the same radiocarbon age on the two fatty acids. And if we do measurements where these two are different and not statistically identical, we can know there was something wrong with the pretreatment and dating of this. So we actually have a control on the date we obtain, which is something we don't really have for any other materials. So this is really a good thing as well. So to do this isolation, I've worked with preparative capillary gas chromatography. So it's a normal gas chromatography instrument that allows to separate different lipid compounds, but instead of sending everything to a detector, there's a preparative fraction collector that is linked to the GC, and this fraction collector has an effluent splitter with different tubes, so we can actually isolate single compounds in single tubes based on how long they spent in the GC. So this kind of instrument has been used for radiocarbon dating since 1996. It was introduced by Tim Eglinton Group, but it was used up to now for environmental samples. So lipids from sediments, aerosols, things like that. And it was in the aim of using radiocarbon as a tracer rather than having an absolute date. So it was not used for archaeological samples before, and it's actually more tricky to use it for archaeological samples because we want absolute dates that we will convert into calendrical age. So if there's any contamination associated, we will bias our age, whereas when you use radiocarbon as a tracer and you follow the evolution of the measurement, it is not as much as a problem as for archaeological samples. So there were a few studies done in Bristol in the year 2000 for actually dating archaeological pottery using single dating and the preparation with preparative gas chromatography. But these studies, there's 3 of them, 2 in radiocarbon in 2001 and 2003, and 1 in antiquity in 2008. And the study were promising, but they didn't reach the accuracy we were needed. So if we see here from this figure, it actually compares the C16 and C18 dates. And like I said, we should obtain the same age from both. and we can see that actually a lot of dates are actually deviating from the 1-to-1 ratio, which means the dates are not always identical for all the dated vessels. There's a few that were well dated, but the other ones were not consistent. So this suggests there was some methodological issue. And in this figure here, they compared the radiocarbon measurement of the C16 fatty acid only and compared it to the non-age of the sites where the pottery came from. And what we can see, so the dates needed to be as close as possible to 0. There was actually a lot of variation. Some of the dates were younger than what was expected. Some of the dates were older. Some were just on the edge of the right age. So there was too much variability to be actually a good method. So there was methodological issues that remain. And then there's a last study in 2008, which only dated one site from 2 pottery vessels, and the age of the site was known by dendrochronology and it was precise to 1 year only, which is age range we see here, the little line, and all the radiocarbon dates were actually younger than what was expected. So this was the state of the art when I started my PhD and had to work on radiocarbon dating of pottery vessels again. So the first thing I did was to try to find all the sources of contamination we could have in the procedure to actually remove them and have much more control on that, because the previous study suggested there was some contamination remaining from different sources because the dates were either too young or too old. So I worked on that, and I took papers that tells all this identification and removal of contamination sources that were published in Analytical Chemistry. So in summary, the first contamination source is what we call the column bleed from the GC column. So the GC column is coated with a stationary phase that contains some carbon. It's called methyl siloxane polymer, and under the heat, this polymer degrades, and some degradation product can actually be associated with the sample at the end. So what we did is use the newest generation of column with low bleed technology, and we actually directly quantified how much carbon was associated with this bleed. And the figure here illustrates actually the amount of carbon related to how much age deviation we would have from them. So we have in average 1 radiocarbon year deviation, which is way below the contamination limit we can use for radiocarbon dating. So column bleed is not an issue with such column technology, which is a good thing. The second point I worked on is what we call the incomplete removal of solvent. I'm sorry if it's not so clear here, but I'm going to talk to you through it. Traps that are usually associated with PrEP GC are this kind of tubes, what we call tubes with a U shape or T shape. So basically, this part here is a part that is linked to the fraction collector, and you have the samples accumulated in this tube. But to recover the samples after, you need to inject some solvent to recover it and transfer it to a vial. And you use an organic solvent which can contain carbon, so you have an isolated fraction which is not contaminated, and then you had an organic solvent with carbon, so you can actually contaminate it again if you don't evaporate well enough the solvent. And it's really difficult to judge with the naked eye because you need to remove it with a nitrogen flow. And if it's too strong, you're going to lose some of your sample you isolated. And if you don't remove enough the solvent, you will have residual solvent that will contaminate your samples and then bias your age. So what we did in Bristol is completely change these new traps and use the new trapping system that we call the solventless trap. So it's just a simple glass tube in which is fitted with glass wool. So glass wool doesn't contain any carbon, and the compounds can be accumulated in the wool and then recovered by just pushing the wool out of the vessel for combustion. So this way, there's no solvent at all, and this source of contamination is removed. And the last part was the memory effect in the instrument in between two samples. So you need to actually, once you have worked with the sample, really clean the instrument. And one point that was causing problem was the preparative unit with a fraction collector, because the part that is linked to the GC was heated to high temperature up to 300 degrees, but the capillaries that goes in the tubes are in room temperature. So there's like a gradient of temperature and accumulation of some analyte at the end of the capillaries. So there's a little fraction that didn't go into the tube. And it actually needed to clean this capillary to not have any memory effect or crossover contamination. So to remove this effect, we just implemented a new procedure to clean this capillary, use a heat gun that just allows to evaporate the residual sample that is in the capillary, and then there's no more memory effect with that. So these were the three main changes to the method to obtain really purified fractions that we can then work with for radiocarbon dating. So when I developed that, we first tested the method with modern fatty acids. So I actually compared direct dates of a fatty acid I bought commercially. So it's what we call the offline measurements. I isolated them in the old tubes that we call the U tubes, and the STS is solventless trapping systems, the new tubes we develop, and compare the dates. And what we could see is with the U tubes, there were a lot of variation in the radiocarbon dates. They were not consistent of the replicates, and they were not always consistent with offline measurements, whereas the measurement with the new solventless trapping system are much more consistent for both fatty acids and actually matching way more the offline measurements. So it really shows the improvement of using this type of tubes rather than the ones that were used with this instrument. And we did some stats on this, all these replicates, and they were all statistically consistent, and the dates between the offline and the trapping system were also statistically identical, which proves that the new method was working well with these modern fats. Then what we did is actually try to validate the method with archaeological fats, and in order not to waste pottery that are quite precious, I worked with what we call bog butters. So if you don't know about bog butters, I incite you to go look a bit through them, because it's really nice archaeology. It's some kilos of animal fats you can actually recover from pit bogs in Ireland, and there's a few in Scotland as well. And currently in the National Museum of Ireland, they have more than 500 specimens they've recovered. And the oldest ones date from the Bronze Age, and the most recent ones are from the Middle Age or even post-medieval period. So it was really amazing to work with that, because we're in presence of kilos of pure animal fats. When you look at the lipid profile of this fat, we have mainly the C14, C16, and C18 fatty acids. So I could work on isolating the two C16 and C18 fatty acids, and I could work with fats with really different ages. So what I did is select 6 fats from the Bronze Age to post-medieval ages, and I did some bulk measurements that I took in the middle of the butter to be sure there was no contamination associated, and compared the bulk dates with the compound-specific dates. And what we see is that there's a really good correlation between all the dates that are dated in individual fatty acids and from the bulk material. It's the same represented here. We have the bulk measurement, which has the dots, and the compound-specific measurements that has the diamond. So this was a way of validating the method with archaeological fats without going into pottery at this stage, in order not to waste them. And from these archaeological fats, during my postdoc, we actually created a new standard for radiocarbon dating from this bog butters. So we actually selected 2 bog butters. This one on the left is from the Bronze Age, and this one on the right is post-medieval. and I was allowed to have about 400 grams of each bog butter. And what we did is clean it and remove all the things we can have from the burial environment, but also animal hairs that were still stuck in the butter. And I cleaned them and homogenized them. So at the end, the standard looked like that. It was divided into 8 pieces. So they are wrapped individually and stored separately to avoid any contamination of the pieces. And we launched last year an entire comparison exercise with 4 other laboratories that are trying to do compound specific measurement using the method we developed. So it's quite a big thing to be able to create this new standard and launch exercises with other labs to really check the dates we have with them and make sure everyone gets the same answer. So to go into the pottery vessels, what we are really interested in doing. First, the procedure is to go and extract the lipids the same way as we do for lipid residue analysis, except that we work with a bit more clay than with lipid residue analysis, but the amount of pottery we take depends on how much lipid was in the pottery. There's sherds I can work with 1 gram, and some sherds I need to work with 10 grams of clay. It really depends on the preservation. Then the lipids are isolated in the preparative capillary gas chromatography, and I work with about 200 micrograms of carbon that I want to isolate with this instrument. And then we go into graphitization to prepare the sample for radiocarbon dating. And then it's measured with an AMS, an accelerator mass spectrometer, to measure the radiocarbon. And this was done on my CADAS instrument that is in Bristol. So now I'm going to go into just talk about radiocarbon dates obtained from pottery vessels and application of it. So the map here presents basically the country of origin of all the pottery materials I dated. So there's quite a lot in Europe, some in Africa, but also in Asia. I'm not going to talk to you through all of these examples. That would be way too much, but I want to give you quite a good overview. First, the method was tested on sites with non-age to make sure we obtained the right age. So we selected sites that are well dated with natural chronology, some radiocarbon dates, and also modeled of radiocarbon dates based on the stratigraphy of the site or the seriation. I worked with sites in different environments, really wet environment, arid or temperate, to make sure the method was working for all kinds of burial environment. And then there's a lot of diverse application on going to date specific food products that are in the pottery vessel, like dairying, marine products. Or I have also example of sites that were not datable with other conventional materials. So pottery vessel was the only way to date the sites, and also some age to have a control on the residue, the results we have from the residue analysis, because sometimes it doesn't really match what we are expecting. And across all these sites, the oldest pottery I dated are from 6,400 BC, and the most recent ones are actually ethnographic sherds, so really modern. So we have also quite a nice panel of ages there. So first, I'm going to present to you the sites that were used to validate this method. So there were the sites with non-age, and they are represented here by the stars. So the first site I dated was the site of the Sweet Track in the UK that was dated by dendrochronology. This site I actually selected because it was one of the sites that were that was used in the 2000s studies that were done before. So it was a good crash test of the new method to see if I obtained different ages at what was obtained about 10 years before I started doing that, and see if we actually this time get the right answer. So this site is Trackway in south of England, and it is in a really waterlogged area. It was constructed in between the year 3807 and 3806 and was in use for less than 10 years. And there were a lot of pottery vessels that were recovered along the trackway, and we can find almost complete vessel profiles. So I sampled 2 pottery vessels that were dated before. So we have in blue the expected date corresponding to the year under dendro. In red, we have the dates that were obtained in the 2008 study, which were all younger than the expected age. And in green here, we have the ages I obtained with the new method. So what we can see here is from the study before, it's actually really consistent ages and older ages than what was obtained, and is actually consistent with the expected age of the site. So it really proves that the new preparation method improved the radiocarbon date we obtained with it. And this graph here actually represents the radiocarbon ages on the 2 ceramic, which are SW1 and SW2, and we combined the radiocarbon age of the C16 and C18 fatty acids because they are statistically identical, and we compared them with tree states that cover from the calibration curve that covered the year 3807 and 3806, which is the age of the construction. And the model really shows that we are statistically identical with this. So we have well dated the site with the new method. So this was a great improvement because this site was attempted twice to be dated before, and this time, on the 3rd time, there's the right answer is finally coming. Another site I used was the site of Çatalhöyük in Turkey. So there was a lot of radiocarbon dates available for this site, and there was a Bayesian model that was built based on the radiocarbon dates. Sorry, there's a typo here, and the stratigraphy of the site, which look like that. And basically, we used the model that was existing, and we added 4 dates on. Sorry, I dated 4 pottery from the site. And so we have 4 new radiocarbon dates, and we incorporated them in the model in the position related to the stratigraphy of the site and the levels where the pottery were coming from. So you can see in green, I highlighted where the new pottery dates have been introduced, and the A and the number reported here is what we call the agreement. And in such model, this number needs to be above 60 to make sure which means the radiocarbon date goes well in the model. If it's below 60, it means that the date is actually not compatible. There's not a good agreement in the model, so it means that there's something wrong with the date you have. And here they incorporate very well in the model, and actually the model didn't change at all. The output of the model for the start and the end of the site is really similar. We just have an average of 4 years difference in between the two, which means that the new radiocarbon dates are compatible with the other one we had for the site. And, another site I worked on was actually a cultural phase, which was Middle Neolithic cultures in Alsace, and there was seriation studies that were done in this region in between all the Middle Neolithic cultures, the Inkelstein, Großgartar, Planning-Friedrich, and Rosein. And so this seriation study was done based on the decorative motif of these ceramics, and I had the opportunity to date ceramics from the Großgartar culture only. And the ceramics I dated fitted well in the seriation study, and again I dated 4 of the vessels, and we incorporated them in the regional model that was this time built with seriation studies that were made on the pots, not any stratigraphy. And again, the dates on the pottery fitted well with the previous model, and we have only difference of the output about 6 years with the site with the original model, so means the dates are compatible, and again, this is well dated. So this was a few case studies to really prove the method worked well, and based on that, then we could go later on to new application and use of this method. Sorry, there was another example I forgot to say. It's a site in Libya, and this site was selected because we are in an extremely arid environment, and in Lucent, where there was floating stratigraphy, if I may say. And we actually went to date the dairy residues that were in the ceramics from this site. And they were from the middle pastoral activity of the site. And we have in green here the dates from the dairy, and in black the other radiocarbon dates from the sites. And we can see the dates are nicely fitting as well. And there was actually one pottery vessel that was dated before on the bulk organics residues that were extracted from the pottery, and my dates on the same pottery but as a compound specific approach was actually way younger than the other date, and actually this date fits in the model when the bulk date was actually too old for the middle pastoral activities. So we again improved the dating, and we can have information on how old dairy was at the site. And also insight into the decoration techniques, because we have really decorated, nicely decorated pottery, so we can have information related to that as well. Now going into case studies. I dated sites that we couldn't date with conventional materials. So there's first the example of the Neolithic origin of the city of London, and also some ethnographic sites. So first, the site of London. So there's not much material from the Neolithic in London, because the city was occupied by Romans afterwards, so a lot of stuff got destroyed by this later occupation. And back in, I think in 2018 or quite recently, 4 pits were discovered for the city of London, and there was only lithic materials and ceramic in those pits. So the relative dating was easy to make based on the pottery typology, but there was no conventional material for radiocarbon dating at all. So the dating of lipids in the ceramic was the only way to date the site at the site of London. And the typology of pottery was what we call plain ball, which is typical from the south of England. So we have in green here the dates on the ceramics from the site of London, and the other radiocarbon dates here are actually dates from other plain ball assemblages that are outside of London, but from the same typology. And we can see we are in the same age range, but we are actually towards the end of the use of the plain ball pottery for London. And yeah, so this was, we have a precise date for the Neolithic origin of London, and we couldn't have them otherwise. And another study we've done for dating, it was actually in collaboration with Julie, because with Catherine Grillo, they studied some material from the Samburu pastoralist group in Kenya. And the ethnographic study showed that they rely mainly on milk and dairy products, but they are mainly stored in gourds, and in pottery vessels, like clay vessels we see here, they are only used for ceremonial instances and only for meat. So you cannot put milk at all in this kind of vessels. And there were some shards that were collected at different settlements for these pastoralists that are actually changing settlements quite often, and the residues, the shards look like they're on surface contact. There's no archaeological context at all. And the lipid residue analysis that Julie Dunne showed that at one site there was actually a lot of dairy products found in the vessels, whereas at all the other sites it was mainly meat as we were expected. So this result was really controversial and was not possible based on the knowledge we had from the ethnography. So Julie asked me to date these residues, so from a site that had no problem, and we obtained a date in between 2002 and 2004 calibrated age, so it's clearly ethnographic. Whereas for this site that showed a problem, the dates on both ruminant adipose and dairy product actually showed a date way older, and it goes into historic periods. So this was explaining the problem we had with this lipid residue results. It's because it was not associated with a modern Samburu pastoralist. So when you are in context like that, where you just have some sherd fragments without any archaeological context at all, the only thing you can do is directly date the sherds because you have nothing else to work with. So this was kind of a nice example to show this, that also lipid residue analysis, you need to be careful of the results because, yeah, it's not always what you think it can be. And then I have case studies on going into directly dating specific food products. So I worked with dairy products in Europe, but also horse products in Kazakhstan, which are nice case study as well. So the first study I have was to date the introduction of dairy with the first Neolithic farmers in Europe, which are from the linear bond ceramic culture. And the culture was established in between France, Poland, Hungary, and there's sort of different migration ways, and we defined different areas of LBK culture, the ones in the west, in the east, and in the southeast, which had slightly different timing for arrival. And there was also a disparity of the presence of dairy of all the LBK sites. So I only selected the site where we found dairy, and I produced radiocarbon days directly on the dairy products. And we compared them with non-age of arrival of the LBK cultures in the different areas. And what we found is LBK, the dairy dates are entirely compatible with the arrival of the LBK culture, which suggests that they arrived and already had dairy with them. They didn't adopt it and dairy through the ages. And it is quite surprising because, like I said, not all the LBK sites actually showed dairy residues. It was mainly at the extreme range of the migration of the culture. All the sites in between actually didn't show any dairy residues. So it was not so clear they really arrived with dairy or whether they adopted it at different time. Another case study I work with, which was quite nice, is some hunter-gatherer groups in the Lesotho. And on this site is from the early Iron Age, the settlement from this hunter-gatherer. There was a debate that ran for years in between morphological analysis of the animal bones that said that there were some domesticated animals with the settlement of these hunter-gatherers. And the DNA study said that these animals were not domesticated. So there was really a conflict in between these two sets of data. And one of my colleagues actually did lipid residue analysis and found dairy residues present in the vessel, which means that these groups had access to domesticated animals because there's dairy in the ceramic vessels. And in order to make sure that these dairy residues are actually from the same time range as the hunter-gatherer, we did some direct dating on ceramic with dairy. And the dates we obtained showed that they were entirely compatible with the culture. It's actually the dates with a simple line here, LEK10 and SHH42. And here the colored bars actually showed the range of settlement for this hunter-gatherer. So the dates are entirely compatible with the settlement, and actually it is another line of evidence that this hunter-gatherer had access to domesticated animals and dairy, which is a nice result as well, and a really nice case study I had the opportunity to work with. And another way of working with specific food products, I went to date lipid residues from Kazakh pottery because there was horse products in the pottery. So one of the sites is actually Botai, which is well known for, or debated as well, for the early domestication of horses. But the fact is, in all the ceramics from the site, only horse products were identified in those ceramics. A few were ruminant adipose or dairy, but most of them are horse products. And what I did is I was able to date 4 pottery vessels with horse products, and I compared them with all the other dates from the site. So we have in red dates on the pottery, and the other ones are the dates available from the site. And once again, we have really good agreement with the new dates and the pre-existing dates. And we can actually have insight into the start of the settlement and consumption of horse products. At this site, we started about 3,600 BC. And there was another site I dated, which is the site of Bestanak. For this site, there's no radiocarbon dates at all. And again, there's horse products in pottery. And the typology of ceramic suggested a Neolithic origin, but there was no radiocarbon dates to confirm. And the dates on the ceramics actually provided date from the 6th millennium BC, which confirms the ancestry of this horse product to the 6th millennium BC. But whether these were domesticated or wild horses, this is another story. But horse products were exploited and used, processed in pottery vessel from the 6th millennium BC in Kazakhstan. And finally, with the exploitation of specific food products, I want to talk a bit with marine resources exploitation, because it's not as easy as dating as terrestrial fats. The problem with marine fats is what we call the marine reservoir effects. Every organism that lives in the water will usually exhibit an older radiocarbon age than terrestrial organism that has the same age. So when we work with marine resources, we need to be aware of marine reservoir effects and correct for them. So we can't have directly a date on this kind of product. And I worked on a site which is Bourne in the UK, which is an isle in the north of Scotland, and it is a Viking settlement. And there was a lot of radiocarbon dates available and a lot of knowledge of the system strategies there. And the dates from the terrestrial animals were about 700, 800 AD, whereas the dates on the marine organism from the same archaeological context were way older, in between 400 and 500 AD. And the lipids in the pot actually provided age right in between the marine products and the terrestrial products, which means that we had a mixing of both marine and terrestrial resources in the pottery. So we need to correct for the marine reservoir effect, but also know how much marine product was present in the fats in order to correct it. So this was like a site I studied because I knew there would be issues with that, and actually develop a methodology on how to really correct, and if we can reliably correct for this marine reservoir effect when we are in this situation. So we have actually information about marine exploitation through lipid residue analysis. So all the stars represented on this diagram is actually lipid residues that showed the presence of specific aquatic biomarkers. And the stable carbon isotope, like you can see, are really shifted from the modern reference ellipses, which suggest either mixing in between the ruminant products and porcine products or the marine fats. But for this site, there's not a lot of pigs, it's mainly marine products. So it suggests that even the sherds where aquatic markers were not detected could actually be present reservoir effect. And I used this stable isotope, carbon isotope analysis values to quantify how much lipids from the how much marine lipids and terrestrial lipids I had in my pot to be able to correct for them, because what I saw is the more enriched stable carbon isotope value we had, the older the dates were, so the more reservoir age I had to deal with. So it looks like there was really a relationship in between the stable carbon isotope values and the radiocarbon age. We suggest we can use this information to quantify marine reservoir. So it's a bit complicated, but I hope I'm not losing you, but it's really important to know we have problems when we deal with marine products, so that's why I want to tell you about it. So what I said, like I mentioned, is to quantify how much marine and terrestrial lipids I had using mixing lines in between the modern reference ranges of marine and terrestrial animals. And I quantified about 30% of marine products in my samples, and I actually compared with radiocarbon age I had, because I was also able to quantify how much marine products I had based on the shift in between the marine and terrestrial bonds. I had two ways of calculating here the marine products, and I obtained similar results with an error. And when I use this value to correct for the marine reservoir effect, I obtained dates that are entirely compatible with the terrestrial age range. So the corrected ages are here in orange and blue, and you can see they are really compatible with terrestrial animal bone, which means we can accurately correct for this marine reservoir effect. It's a bit more work, but it's possible. If you can avoid working with marine products, that'd be great, but sometimes we don't have the choice, and we can do that. And another site I had a problem with is the site of Cliffsand Farm in the UK, which is by the British Channel. And I actually dated sherds that were dated before by the charred residues. And all the sherds I had from the absorbed residues were older than the surface residues by up to 150 years. And my dates were actually not compatible with the site chronologies. They were too old. And it was really strange. We didn't have the same age for the surface and the absorbed residues. And based on the stable carbon isotope values and other information from the site, there was no possibility for me to have pigs in the pottery because the pigs were non-existent, and I had values shifted from the ruminant ellipses towards the pig or the marine range, and there was no biomarkers for marine products. So actually, this age supported that I had a marine reservoir effect in my dates as well that we didn't have from the surface residues for some reason. And I actually had to correct for them, and I obtained these are the dates in green, the corrected age, and the surface residues are the dates in blue. And what we found out is actually radiocarbon dating can be used as a tracer to identify the processing of marine products in the pot when we don't have other line of evidence, because there was no fish remains or marine remains at the site. There was no evidence from the biomarkers in the pottery, and it's only the radiocarbon dates that actually told us there was some marine processing here. So this is also a nice study, and it got published just this year. Yeah, sorry, I should have shown you that. That's what I was illustrated when I said the stable carbonized values are enriched towards marine and non-marine fats. And we have mainly sheep, cattle, and a bit of horse at the site and no pigs. So yeah, compound-specific radiocarbon dating provided the evidence for the aquatic resource processing when we didn't have another line of evidence. So I'm finishing here, so I hope I didn't lose you in all this case study, but we have a new method that has been proven successful for a lot of sites, different age range and burial environments. We can safely and work with terrestrial animal products, and we can work we can use this method at sites where we don't have other material for radiocarbon dating. This provides an alternative way of providing radiocarbon ages, and we can directly date specific food commodities, and we can actually also date directly typochronologies and well, refine the relative typochronologies with radiocarbon dates. The limits of the method, like I mentioned, is when we have marine product processing, we need to deal with reservoir effects, so something we need to be aware of. And sometimes we may have potteries that are not fitting the age we were expecting, so we need to be careful with materials that can be intrusive or residual in the context we work with. And that is why working with refitted vessels or vessels that are decorated and typologically characteristic will provide more assurance that you are dating a material that is well associated to the context it was recovered in. So when you have a big stratigraphy like in Çatalhöyük, this may be of interest to not work with a single portrait, but a portrait that come from a vessel from which you have found different fragments. And another limit I didn't mention, but it's when you have a big pot, you may not have homogeneous lipid preservation within the vessel wall. You can have more accumulated towards the top or towards the bottom. So it's better to work with lipids that are preserved in high concentrations, because when you work with low preservation, if you need to sample 10 grams of pottery, you may have issues of extracting enough material for dating due to this inhomogeneous partitioning of lipids. And I'm still working with this method and have some perspective is to work with smaller sample sizes when organic lipids preservation is an issue at some sites. So we can try to work with less material, but also radiocarbon date other class of compounds that we can find in pottery. Julie showed you a nice example with plants where you have a lot of alkanes, and there's aim to go and date these other compounds other than the fatty acids to be able to date other food commodities as well. And I thank you for your attention. I'm going to stop. Yeah.
Thank you so much, Emmanuelle, for this really very interesting presentation. I'm pretty happy before starting this session of questions and answer. Pretty glad to see the number of attendees. Actually, we have been over 60s at some point. Also quite well attended regionally speaking, because I mean, we have obviously the majority of attendees from Italy, but I've seen people from Egypt, Tunisia, Algeria, so North Africa very well represented, then Poland, Romania, United Kingdom, and Andorra. So really, I'm very happy that these talks you gave had such impact also internationally. I had no doubt considering your names, so I'm really very happy for this. So I would like to get a question from all of you. I'm sure that there will be question for Emmanuelle and Julie, so please feel free to who wants to break the ice. Federica, please.
Hello, hope that you can hear me.
Yeah, very well.
Switching on my camera. Thank you for this very fascinating talk. And what I would like to ask is, which is the minimum number of samples for obtaining meaningful results and for both kind of analysis? And whether it would be better to sample sherds and which part, I mean, rims, walls, bottoms, or all vessels? Thanks.
Shall I start with the sampling and then hand over to Emmanuelle for the radiocarbon sampling, because they are separate things? Yeah, so if you're going to look at an assemblage, we recommend a minimum sample size of 30 sherds from different vessels to get a statistically reliable data set. And if that would apply, say, for example, if you're going to look at one vessel type or one chronological period. But if you wanted to make comparisons across chronological periods, say, or different vessel types, we would say 30 for each, or say 25 to 30 for each. In terms of what part of the vessel to sample, we normally ask for rim sherds because, you know, I mentioned earlier about the pot and the lipids floating on the top of the vessel. So we find that preferentially they absorb and are found in greater concentration in the rim area, but are still found in the body and in the base. So if you don't have rim sherds, then body sherds are the next best thing. Obviously, that might not, although that would apply to cooking pots, it may not necessarily be the case in terms of specialist vessels. So, for example, roasting pans, you might get more in the base, but we would discuss that with you first. I think that covers the sampling for pots. I'll hand over to Emmanuelle because it's different for radiocarbon dated samples.
Well, actually, for radiocarbon dating, as I said, we first need to do the lipid residue analysis to know which sherds have lipid preserved and what is actually in the sherds. The number of samples that you can date from site will be quite variable based on the preservation, but it's about 10% of an assemblage. So it depends how many radiocarbon dates you're aiming for. But if you're aiming for 3 radiocarbon dates, and 30 portrait is a good number to look at. But if you're aiming for more dates, you may need to look first at other higher number, but it's really going to be site related. So
thank you. If I can add another small question, if it is possible also to work on materials from museum collections instead only on material that's coming from excavations.
No, yes, I mean, absolutely. And in fact, by far the largest amount of analysis done at Bristol has been carried out on museum collections. The lipids are preserved in pots for thousands of years. So it doesn't particularly matter, you know, excavation and then storing in a museum for 10 years or up to 100, as we found in doesn't necessarily, it won't have any impact on the lipids which are preserved within the fabric. Obviously, best scenario is freshly excavated sherds, but we know that's not always applicable. So,
well, I wanted to add that you need to be careful of the storage of your portrait, but because with museum collection, often we see a lot of plastics in the residues that come from the storage. So if you can avoid plastics to store your pots, then do it, wrap it in foil or things like that, especially if you want to carry on this kind of analysis later, so you avoid this kind of modern contamination.
Thank you.
Thank you, Julie and Emmanuelle. Do we have other questions? Yeah, Adelaide, please.
Can you hear me?
Yeah, very well.
Okay, I would like to ask you a small question to Professor Dunne. I would like, first of all, to thank you for your inspiring presentation, and I find it particularly fascinating And I have a question about methodology. In particular, once we define the profile of the lipids that has been absorbed by the vessels, and we know if they derive from animals, plants, or maybe diary, is it possible at this point to broaden the spectrum of the analysis and maybe identify, for example, the geographical provenance of the plants that were processed by the on the vessels, or maybe this kind of analysis ends in itself?
Yeah, thank you for your kind words. I think what we can do when looking at plant processing in pots is determine whether the plants were either of C3 or C4 origin. So that is in itself a geographical indicator, because, so for example, in Neolithic Europe, only C3 plants were present because C4 plants aren't introduced until something like, I think it's the later Bronze Age. So that is a geographical indicator in itself. And the difference between C3 and C4 plants is also an environmental or ecological indicator, because you might find, for example, as we found the ethnographic sherds that Emmanuelle mentioned from the Samburu people in Kenya, those lipid signals were very, very heavily C4, reflecting the very dry and arid region that the pots were used in Kenya. So they do provide both an environmental and a geographical signal. I mean, obviously, you would assume that plants processed in the pots were from a fairly close area. So that would, as I mentioned about tying together the and the lipids, we also marry together any archaeobotanical remains there may have been, such as at Takakori, there was a vast amount of archaeobotanical remains, although not many in the NOC project. But that does also give us an indicator of what may have been processed in the past.
Thank you very much.
Does that answer your question?
Yes, thank you.
Do we have other questions? Don't be shy. I have a question, actually. What always intrigues me, I mean, I give the question to both of you. Is this an issue concerned with the recovery rate? Because, I mean, since you had the experience really and the possibility to analyze so many samples, so many sherds, could you find any particular trend in recovery rate that can follow a particular pattern linked to, I don't know, particular condition of the soil, of the deposit, environmental factors? Because and Julie, you know what? Sometimes I'm really struggling to understand. I mean, in our case, for example, you had the chance to analyze both the pottery sherds from Dukanet El Kutifa and Kefamda, which are two sites, just one Neolithic and one Capsian, just 5 kilometers apart from each other, and one with the exceptional, good recovery rate, and the other one with very low recovery rate. So this is quite interesting to me to understand better.
Well, I think the answer is that it isn't fully understood. So if we do know, for example, that certain preservation conditions are better than others. So, for example, acidic soils, which don't preserve animal bones or human bones very well, do preserve lipids very well. But other conditions, such as arid conditions, for example, in the Libyan Sahara, lipid recovery was excellent there, and that is normally a good indicator. the case of Dunne is a bit odd in that, yeah, the recovery was very different from those two sites. And it may be that that is actually not site dependent, but perhaps more related to use by the different groups of their vessels. And we don't really know the answer. I don't really know the answer to that. But yeah, I mean, there is a real difference in lipid recovery, certainly across African contexts. I think I think in European contexts it tends to be a bit more uniform. Would you agree with that, Emmanuelle?
Yes, but I also see shards in a really close area. So one site had a really poor lipid preservation, and the other one were all really great. So yeah. But we see also a difference with typology of vessels. So usually the pots that are assumed to be cooking pots usually have a lot of preservation, the highest rates, when the pots that are more refined vessels or for serving dishes usually have lipids, but in way lower concentrations. So the function of pottery also influences recovery.
Yeah.
And usually, Emmanuelle, is consistent the rate of preservation of that Julie has in recovery lipids with possibility to have good results in dating?
Well, if you have enough lipid preserved, then you can have good radiocarbon dates. If the dates are not good, it means that maybe there was some compound that you could not separate from the fatty acid, so it's more like a methodological problem. But if they are preserved, there's no reason why they can't be dated.
Okay. So we have another question from Alessia.
Can you hear me?
Yeah.
So thank you for your presentation. It was very interesting and enlightening. I have a question for both, and I hope it's not my field, so I hope it's not a stupid question. I'm sorry. So while you were presenting these approaches applied on ceramic vessels, I asked myself if this approach was ever applied on lithic paraphernalia, like in pre-ceramic archaeological context, and if it was fruitful as it was on pottery. And if yes, which kind of stone material was much preserving for the residues?
Yeah, it's an interesting question. So one of the reasons lithics, it's always been thought that one of the reasons residues preserve so well in pottery is that they are protected within the ceramic matrix. And what that also does, of course, is ensures that you can be clear that there's no contamination, no modern day contamination affecting results. Lately, we have been asked to do analysis of a uniquely, I think, British and Irish feature called burnt mounds, which are sort of, well, they're ubiquitous in Ireland and quite common in Scotland as well, similarly to the bog butter. And they're these massive piles of mounds of burnt stone where people think either animal cooking or boiling water or bathing, other technological things are going on. So we've done some analysis of those and also some analysis of Anglo-Saxon burnt stone pits, which are 6 foot long pits, just full of highly heated fire-cracked stone. And we have got very nice, much to my surprise, lipid profiles from these, and they are indicative of ruminant carcass processing. So processing probably either big joints of meat or whole animals. The results have been better from the Anglo-Saxon pits and less good from the burnt mounds, but they are still yielding results. And I think that's because people are sampling from within the pits. So the lipids are preserving. We've done some microscopic analysis. of the stones themselves, and they're very cracked, so the lipids are probably preserving in those. But in terms of, I think you really mean more individual lithics, don't you?
Yeah.
Yeah. I think the issue with trying to do it for that is that is firstly, well, mainly the contamination issue, because how could you be sure that what you were getting from it was archaeological as opposed to modern, because you get finger lipids on your hands, and so lithics which are bound to be handled several times, if they were to yield lithics, we would have no way of determining whether they were modern or archaeological. And the other issue, I think, would be abundance. You know, how much would be preserved? I think it would be such a small amount. So on the whole, I think there's a good reason that residue analysis of lithics themselves hasn't, you know, gone very well in the past. But there is potential for larger groups or clusters of stones, I think.
Thank you.
Thank you so much, Alessia, for your question. Do we have other questions? If you feel more comfortable, you can also do it in Italian or in French. Just feel free to ask if you have any doubts, curiosity, you want to know more about some aspects. No, we don't have anybody. So thank you so much, Julie and Emmanuelle, for your generosity in staying with us this afternoon. It was really a pleasure to listen your really fascinating presentation, and to see also the variety of study cases, how these methods can be really applied to different kind of context, geographical areas, different periods, and this is really very, very important to see this transversal approach that can give us so much information, and can be so helpful to answer proper archaeological questions. So thank you so much once again, and hope to see you soon around the conference in the field somewhere. Thank you, grazie mille.
Yes, thank you, everybody.
Thank you.
Thank you so much for the interesting information. Thank you very well.
Grazie mille. Buonasera, arrivederci a tutti.
Arrivederci.
Grazie, arrivederci.
Salve, grazie.
Arrivederci.
Thank you so much.
Julia, shall I stay on? I think you wanted to ask me something about.
Oh yeah, why not? Yeah, thank you.